that many of you are probably wishing you were outside somewhere. So thank you for being in front of your computers and tuning in tonight. I'm Heather Smith from the Watershed Center, and Caroline Keeson from Tip of the Mitt Watershed Council is co-hosting this training with me. Can everybody see my shared screen?
Yeah, I can see it.
Great, thank you. Okay, really quickly, I'm going to go over our agenda and some housekeeping. What we're going to do first is introductions. Then we'll go over our COVID safety protocols in light of kind of the new situation that we're faced with in 2020. We'll review the MyCorps program and the monitoring programs developed by MyCorps. We'll talk briefly about aquatic invasive species decontamination and ensuring that we are not spreading invasive species in our stream sites. We have a special guest speaker, Dr. Jo Lattimore, and she'll talk about invertebrate adaptations, and then we'll conclude with group breakout sessions for both Tip of the Mitt and the Watershed Center, where we'll go over some specifics about this monitoring season for each of our organizations. So briefly, some housekeeping information. This meeting is being recorded and it'll be available at a later time. So if you would like to review the meeting again, you're welcome to do so. We'll send out that link once it's available. We're going to ask you to please use the raise hand function to ask a question if you have one, and there'll be time for questions certainly at the end of Dr. Lattimore's presentation and during our group breakout sessions. And then I'm going to ask that you please mute your microphones and please turn off your video during the presentations. That will hopefully help us run a little bit more smoothly today. Okay, so I'll start. I'm Heather Smith at the Watershed Center, Grand Traverse Bay. I am the Grand Traverse Baykeeper, and I coordinate our volunteer stream monitoring program that we call Adopt a Stream. So I see a lot of familiar faces, and I'm happy to be here. And I will next introduce Caroline, co-host from the Tip of the Mitt Watershed Council.
Hi, my name is Caroline Keeson. I work for the Watershed Council in Petoskey. So our area is just a bit north of Heather's, and we have stream teams all the way to Sheboygan. We have a couple other volunteer teams or volunteer programs that I oversee too, and then we also do a lot of our own stream monitoring. So I'm happy to see some different volunteers and get to meet people from a coordinating group.
And then very briefly, our special guest tonight is Dr. Jo Lattimore. She's an outreach specialist in the Department of Fisheries and Wildlife at Michigan State University. Jo, do you want to briefly say hi?
Sure. Hi, everybody. Again, Jo Lattimore here from MSU. I'm actually, although I have an office in East Lansing, I live and work out of home in Traverse City and have done that for the last 4 years. So I'm a Northwest Michigan person myself, and I'm excited to be with you here today.
All right, awesome. Thank you for that. And I think what we'll do now is go around and have each volunteer introduce themselves very briefly. Maybe you could state your name and whether you're with Tip of the Mitt or the Watershed Center, and then maybe your favorite macroinvertebrate or your favorite stream site, maybe the stream site that you monitor. We'd love to just hear from you for just about a minute, just so that we know who's in this presentation with us tonight. So why don't I start by calling folks out. Michaeline, you're right under my, you're the first image that I see. So do you want to briefly introduce yourself?
Sure. I'm Michaeline Horton. I'm with Grand Traverse Resort and Spa, and we are with the Watershed Center. We test and sample the Acme Creek sites. There's 2 of them. One we haven't done for a few years, but I'm going to tell you that's my favorite because it's the only one I've really done.
Awesome. Thank you. Let's see next. See if I can go through everybody. Jeremy.
Hi, I'm Jeremy Chambers. I live up in Petoskey. This is all 100% new to me, but I'm tucked in there by Walloon, so I'm guessing they'll probably want me taken to one of the streams near me. But I'm really excited to be here, so thanks for putting this on.
Great, welcome. And Tanny?
Hi, Tani.
Tani, sorry.
That's okay. I've only done this once, and we weren't able to do it for the spring, so I just did it last fall, and I did it with Heather, and we picked a stream that is maybe about 2 miles south of Almena, and it's a very small stream, but we found really good organisms. Actually, Heather found really good organisms.
Hey, you helped.
Oh, well, maybe I got them in the water, but yeah, no, I couldn't identify them. But thank you very much.
Yeah, well, welcome. Nice to hear you. And you are on Weaver Creek, I believe.
Yes, Weaver Creek.
And Joan? Oh, we'll have to unmute.
There we go. Joan Bonney with the Watershed Center in Traverse City. I monitor with Frank Simpkins, excuse me. We've had fun doing that for a few years now. We sample a site at Kids Creek near the Hospital Heart Center. I'll let Frank tell you about the other site. Anyway, I'm trying to think of my favorite macroinvertebrate, but I can't. I like them all.
Great. Let's see. Emily, would you like to introduce yourself?
Sure. Hi, everybody. I'm Emily. I actually coordinate the stream monitoring effort out at Grass River Natural Area. So we are monitoring Cold Creek this Saturday, actually, which flows into Grass River in the Antrim chain of lakes. And yeah, I thought I got sent this invitation for this from one of your volunteers, and I thought I'd pop in and it'd be a nice refresher right before we go out and monitor this Saturday. And I also wanted to hear from Joe, so thanks for having me.
Thanks for being here. All right. How about Luke?
Hi, can you hear me?
Yes, we can hear you.
Okay. Sorry, I joined a little late, so I missed I missed kind of what we're doing, but I've listened to the last few people, so it sounds like it's just a general introduction.
Yep.
So my name is Luke Fogarty. I just started doing the stream sampling last fall. Me and Heather worked on the Upper Boardman up by Broomhead Road, and we missed it this spring, but excited to head out again.
Awesome. Yeah, that is a beautiful site. Thank you, Luke. And Frank.
I am Frank Simpkins. I also live in Traverse City, and I work for Joan Bonney, sampling part of Kids Creek and Hines Creek, which actually flows into Cedar Lake.
Great. Nice
Cute
to
little
see
creek.
Yes. Nice to see you, Frank. Thank you. Let's see. How about James or Jim?
Hi, I'm Jim Egan. I live in Suttons Bay. I'm part of the Watershed Center. I joined late last fall, and Heather and I never had the opportunity to get out and sample a stream, so I'm anxious to get out and actually start doing something.
Great. Thank you. I've got some ideas, so we'll talk.
Okay, great.
Let's see. Who is next? How about Jay Peterson? Jay, can you unmute and introduce yourself? Oh, looks like you're unmuted now.
I think I did. Okay. Yeah, I'm Jay Peterson. I live here in Traverse City. My wife and I moved here when we retired about, I guess, 6 or 7 years ago it's been. we sampled the stream with Heather last year, Miller Creek, and that was very successful. Unfortunately, I did the creek myself in the fall and didn't get such a diversity of animals. In fact, I have a unfavorite one now, the scud, which is 99% of what I managed to collect. But I must have done something wrong, so that's why I'm here to get a refresher. So, thanks.
Well, thank you for being here, Jay. Let's see, Rod Rebant.
Hi, everybody. I'm Rod Rebant, and I work with Heather at the Watershed Center, helping with the Mitchell Creek watershed, and I also do some work on Carpenter Creek, which is part of the Boardman River system. And I also help Heather on the validation of all of the collections that are done across the watershed. And I am also a board member of the Watershed, and I absolutely enjoy working with the group. And I think I'm somewhere between 8 and 10 years of working with the group, and I'm looking to many more years.
Great, thanks, Rod. Yes, Rod is a set of eyes in the expert entomology team. So thank you, Rod, for all that you do for the program. Let's see, how about Mike Foley? Or Foley?
Hi, Heather. Hi, everyone. I live in Traverse City, and I work with the Watershed Center on the Adopt a Stream program. I've been sampling out at Yuba Creek last year, a couple of different sites, and looking forward to getting back to it this fall.
Great, thank you. Let's see, Jenna.
Hey, everybody. Hi, I'm here in Traverse City, and my husband Nate and I do a section of Kids Creek here, and then I teach. Oh, wait, that was on. Hi, everybody. I'm Jenna Scheib. I do a section of Kids Creek with my husband Nate Winkler, and I teach biology and ecology out at Interlochen Arts Academy.
Great, nice to see you, Jenna. Okay, let's see. Marty?
Yeah, hi. My name's Marty Jones. I live in Mullet Lake Village, and I help monitor Mullet Creek.
Great, welcome. And am I missing anybody?
There's one person I just admitted, Jay Holden. I don't know if we heard from him yet.
Yep, let's see. Jay, could you introduce yourself briefly?
Sure. Jay Holden with the Grand Traverse Watershed. Volunteered last year, have done one sampling and looking forward to doing it again.
Great, thanks for being here, Jay.
Mm-hmm.
And I think that's everybody. Anybody that I'm missing, please speak up. So thank you to everybody for being here tonight. We're excited. I'm going to launch into the presentation now. So our next step is to go over our COVID safety protocols.
Hey, Heather. We're not seeing your actual slideshow. We're seeing your editing window.
Oh.
So I would stop sharing and then start sharing again and choose the actual presentation. It'll probably show up as a separate option for you.
Thank you very much, Joe. How does this work, folks?
That's it. You got it.
Okay, sorry about that, and thank you so much. Okay, so I want to briefly go over COVID safety protocols. Our top priority is really to ensure that our volunteers are safe and comfortable in the stream, that all our staff remain safe, and that the community is safe. So we are asking folks to wear masks while sampling and identifying invertebrates, and that's especially important if you're with folks that are outside of your household. We're encouraging folks to maintain 6 feet or more of space between individuals in different households. We suggest conducting all identification outside in a well-ventilated area. Be sure to hand sanitize tools and equipment before handling between different persons. We are suggesting frequent hand washing and sanitizing. And if you are, most folks are driving to their sites, but we are encouraging folks in different households to ride in separate vehicles. And then we'll please ask you not to sample or pick up or drop off equipment if you feel ill. So we really want to ensure that everybody's safe and comfortable. And if there are any questions, certainly let either Caroline or I know. Okay, so our MyCorps Volunteer Stream Monitoring Program, MyCorps stands for Michigan Clean Water Corps, and it was created to assist the State Water Quality Agency, which is now the Department of Environment, Great Lakes, and Energy. It used to be EGLE, I'm sorry, it used to be DEQ, but now it's EGLE. It was created in 2003 with a mission of collecting, sharing, and using reliable data, educating and informing the public, and creating water resource stewards across the state. It's really a way for the state to get data on a lot of our small wadeable streams that are otherwise left unmonitored. There are about 50 organizations across the state that are participating as volunteer stream monitoring programs. So places like the Watershed Center and tip of the Mitt Watershed Council are kind of the local coordinators, but we receive our protocols from the MyCorps program, and they provide us as local groups with access to their database, training resources. They review quality assurance project plans every year for us to ensure that we're collecting high quality usable data, and of course they're very good at giving us advice on how to best administer a volunteer monitoring program that generates usable data. And so this is no surprise to folks. There are lots of benefits for volunteer monitoring. You know, it's a fun way to get outside and have community members take ownership of their streams and really get out in nature and be present in some of our beautiful stream systems. It's a nice hands-on fun program for folks. We have a lot of different age groups involved, but certainly a lot of school-age children. Again, it allows for more streams to be sampled across the state. Volunteers will learn a little bit about water quality indicators and issues, what we're doing on the land and how that affects water quality, and it provides useful data for a number of uses. We use it to establish water quality baselines across our watersheds. We use it to flag issues. We use MyCorps data to assess restoration projects and also determine where restoration might be most needed. And then finally, our data, because it's entered into the MyCorps Volunteer Stream Monitoring Program, that state database, it's available to state water resources managers that can use it to really assess the quality of our area streams. And now I'm going to turn it over to Caroline to talk about our macroinvertebrates.
All right, thanks, Heather. Can you guys hear me? I'm going to take over control here. Okay, let's see here. Okay, so macroinvertebrates. We actually use a macroinvertebrate biotic index. So macroinvertebrates are what we call bioindicators. So they can indicate if a stream water quality is good or maybe so-so or maybe very poor. So they actually show the scale of the quality of the environment. Oops, did that just, oh, what's going on here? Oh, there it is. Okay. So the index is based on the different types of macroinvertebrates and how many of each type. So if we find a large amount of a very sensitive organism, the stream will skew more towards good water quality. And if we find a large amount of the organisms that aren't so sensitive, the stream will have poor water quality. And we're hoping that Joe explains about some of those adaptations that make macroinvertebrates more or less sensitive to water quality. Okay, I'm going to try to advance this. Okay, Heather, I don't know. Oh, there we go. Okay, so why are we looking at macroinvertebrates? Well, they're always going to be in freshwater streams. They're a good indicator of water quality because we know what their tolerances are. They're very inexpensive to sample for. Once you get the equipment, you basically just need to spend your time out there sampling, which is why we luckily have all of you volunteers. And they don't move very far, so because they live in the water all the time and they're not moving around a lot, we can visit the same stream site year after year and expect that hopefully the same or a similar population is going to exist. Okay, so the things that cause the invertebrates to change over time could be different seasons. So sometimes there'll be more macroinvertebrates during a certain time if there's more algae in the water, like in the summer. They like to live in different areas with dissolved oxygen. So if there's more dissolved oxygen in the water, we might find more stoneflies or mayflies. If there's less, we might find things like scuds. If the substrate changes, so if there's less cobble or maybe more cobble, that means there's different places for those macroinvertebrates to live. So that can cause macroinvertebrate populations to change. And so some of the human influences that cause the populations to change could be too many nutrients, a change in pH, removing riparian vegetation. So that would be, removing native vegetation and possibly replacing it with turf grass, making a channelized stream, basically removing the places for the macroinvertebrates to hide and places where they would like to eat. And then just other in-stream habitat alterations. So road stream crossings is a big one. Dams is another one where you might see differences in macroinvertebrates over time. Okay, so these macroinvertebrates are extra important because they're at the base of the food web and they're really important cyclers of nutrients, and they do a lot of decomposing. So a lot of the inputs into a stream, like woody debris and leaves and vegetation, the stream system relies on those macroinvertebrates to actually eat all those, eat the big pieces of wood and turn them into little pieces of wood. And then eventually the nutrients get into are available for uptake in plants and other organisms. And of course, what we might think is the most important thing is, of course, fish eat them. So that's really good for us anglers. Also amphibians, reptiles also eat them. And then of course, once they become adults, such as a mosquito, a bat might eat that, or even a bird. So they really have a big effect on things in the water and in the air. Okay, so I'm going to talk a little bit about collecting and the best way to collect, because Heather and I know that some of our teams need some help with this. So make sure that when you go out, everybody should have an assigned role. So you should have collectors, who are people who are going to go in the stream and get the bugs. You should have pickers who are assisting getting the bugs out of the nets and to the people identifying the bugs. And then there should be someone recording, filling out your data sheet, and ensuring that all the equipment leaves the site. You should start downstream and work your way upstream. Otherwise, you could accidentally, kick things downstream, and that will change your results a little bit. Try to get a 300-foot stretch of stream if it's passable. So that means if you can safely wade it, go ahead and sample it. If it's too deep, you can skip it. And then you want to try to collect specimens for 30 to 60 minutes and do the same amount of time for sorting and identifying. So that will hopefully give you enough time to get into many different habitats and also spend time looking for the little tiny critters that you might not see otherwise if you just do it quickly. Okay, so here are some examples of the types of habitats that you want to get. So each one of these habitats will have different types of macroinvertebrates. And so when you think back to the index, where we're rating these streams based on the different types of insects and their abundance, we want to make sure that we get a good variety, because that's going to help the stream score. So this is called a run. It'll be a pretty straight part of the stream or the river, and I would say the best feature to look out for is that it is mostly flat on the surface. There's no wood sticking out, there's no cobble, there's no whitewater rapids or anything like that. It's just a pretty calm, maybe not super calm, but it's like a straightaway. Okay. All right, so the next one is riffles. So riffles are marked by cobbles, boulders, bigger rocks. It's an area of turbulent water, so the water might be a little bit white, and that's actually more oxygen in the water. So if you sample where the water is really turbulent, and then a little bit where the water is a little bit calmer but still in the riffle, you can actually get different types of macroinvertebrates just in this one place where your really high quality macroinvertebrates are going to hang out, like the mayflies and the stoneflies. Okay, so pools, and this is actually, looks like a beaver dam that's created a pool. Pools are going to have some different critters in them. Try not to get too much muck or silt in your net. And you're just really trying to sample, you know, a calmer area of the stream. So this might be where macroinvertebrates that don't have the best swimming skills, they might hang out over here. And so this might be on the edge of a stream, not necessarily right in the middle. It'll be an area of deeper water. Okay, so here's some cobble. So cobble has a lot of spaces in between the rocks, and so we call those interstitial spaces, and that's where the macroinvertebrates, they like to hide in there, really close to the bottom. So this is a good place to set your net down and kick a little bit towards the net to try to get some macroinvertebrates dislodged from the rocks. So remember, you're trying to get what's in between those rocks into your net. Submerged wood is another good habitat, and actually you can take pieces of wood out of the stream and take them onto, you know, into the area where your pickers are, and you can have them pick pieces of wood to keep everybody busy. So there's a couple different types of caddisflies that like to hang out on submerged wood, and also it's a good place for little tiny things like black fly larvae and mosquito larvae. So definitely take these out of the stream so that you can see, you know, is there anything living on these. You can also kick into your net near pieces of wood too. They're really big. Aquatic plants is another good place to look for macroinvertebrates. Caddisflies also like this. So this would be another place where something that was slower swimming would try to hang out. And so this might be on the stream edge. It could be aquatic plants or algae. So different types of macroinvertebrates like to live in the green stuff. And then of course, undercut banks and overhanging vegetation. So there might be some roots in the water underneath the edges of your stream bank, and you can put your net under there and kind of shake and brush against the roots and the edge of the stream to capture more macroinvertebrates that way too. Just try not to get too many scuds. That's a good way to get scuds. Rocks is another good place to look for macroinvertebrates. You can also take these out of the stream and have someone who you know, has a little time on their hands. You can have them pick stuff off of the rocks, and then you can just put them back in the stream when you're done. Okay, so let me see if I can, I don't know if I can turn this poll on. Hold on a second. Okay. Okay, Heather, I think I have to give up remote control. Oh, weird. All right, can you all still see that screen? For some reason, I'm... Anyway, okay, so I was going to have a poll, but for some reason, I can't get it to pull up. But
I might be able to get it.
Oh, there we go. Okay, so what kind of stream habitat is this? I'm only giving you 4 options, even though I've gone over quite a few. You should be able to see this poll, and you can go ahead and click on whether you think it's a pool, a riffle, submerged wood, or a run. What would be the best way to qualify this habitat? Oh, it said the polling is closed. Okay, let me try it again. Okay, there you go. Now you should be able to see the poll. Okay, well, I think I did my job because everybody got that it was a run. All right, good job. All right, so there should be another quiz. I don't know why, Heather, but I can't advance the slide. Here we go. Oh, there was one more. Okay, here's the other one. There we go. Okay, so one more. Which stream habitat type is this? Pool, riffle, submerged wood, or run? Okay, we got some different answers here, and I would say that they're both right. We definitely have some riffle and some submerged wood. You could possibly even say there was some cobble in there. So this would be a good place. You'd want to spend more time here because there is an abundance of different habitat types here. Okay, so we'll go back over this one. Okay, so the other thing to think about when you're out in your stream is decontaminating for aquatic invasive species, especially if you are an angler or just somebody who likes to hang out in streams. Make sure that your waders, if you're taking your own, make sure that they have dried out for 5 days. It has to be longer if you have felt bottomed waders, so the lug boots are actually best. And the big concern is the New Zealand mud snail. It's very small and it's hard to see, and it's recently been found in the Boardman and Osceola rivers. So this is a concern both for the Watershed Center and Watershed Council, because most invasive species we can see, but the New Zealand mud snail, you know, could definitely be more of an issue for us, and we just want to make sure that our monitors are doing what they can to make sure that we aren't spreading this from stream to stream. I think that might be, yep, that's it for me.
All right, Dr. Lattimore, are you able to share your screen?
Yes, I am. Does it look good to you?
Oh, it looks great.
All right. So thanks to Caroline and Heather for inviting me. I'm really excited to chat for a few minutes with you about aquatic macroinvertebrates. I've spent the last several years really focused on Michigan's inland lakes. So I do a lot of works on aquatic plants and invasive species in our lakes and so forth. But my background is all on rivers and streams. I did a master's at Notre Dame where I looked at stream invertebrates and how they were impacted by land use and sediment problems and subsequent stream restoration and how they indicated the recovery of that stream. And then for my doctorate, I focused on rivers in central Michigan, looking at their fish community and physical habitat there, and also looking at how disturbance influence fish communities. So it's fun to come back and think about rivers and streams. My first job after grad school was for the Huron River Watershed Council in Ann Arbor. I was their watershed ecologist for 4 years, and during that time got really involved with their Adopt a Stream program. And so I spent a lot of time doing exactly this kind of thing, working with volunteers, getting them prepared to go out and do biomonitoring on the tributaries and the main stem of the Huron River. It was a lot of fun, a lot of excitement. And since I moved on to MSU, I focus more on lakes, but ever since I've worked at the Huron, I've been involved with MyCore. I worked on developing the protocols for monitoring both lakes and streams, and it's exciting to be thinking about bugs again. So for the next few minutes, I'm not going to take up a ton of time, but I just want to provide a little information on some of the kind of fascinating aspects of aquatic insect life. You know, I've taught a lot of volunteers about this. I also taught aquatic entomology, study of aquatic insects at MSU for our undergraduate programs. So I'm going to wrap all the cool stuff from those courses into the next 15 minutes. And the idea, one of the ideas behind this is to maybe give you a little insight on how these bugs live their lives, and that will make it maybe easier for you to find them, because a lot of that is tied to their aquatic habitat and the way they've adapted to life in aquatics. So let's take a look here at some of the fascinating things about aquatic insect life. It all starts with reproduction, right? So that's kind of the surprising thing that a lot of people, when they first realize that many of the insects that they're used to seeing on land and flying around actually have an aquatic juvenile stage. And that's the stages that we're looking for most of these groups. So the midges, the little tiny non-biting gnats that we see everywhere and are their larvae are very common in our streams. They're kind of a great example of one of the typical aquatic to terrestrial life cycles that we see with insects. So you have your flying adults here, the little tiny gnats that fly around and like to fly in your face. And then those mature adults will lay their eggs on the water surface or near the water surface, and then they'll drop into the stream. And then those hatch into the aquatic larvae that we see. You're probably familiar with the bright red colored bloodworms. That's just one group of the midges, the Chironomidae. These are most likely to be found in areas with low oxygen. So more of your mucky, kind of silty streams will have a lot of these more bright red aquatic larvae that we call bloodworms. And they're bright red because they have a compound that's like the hemoglobin in our blood that helps them circulate oxygen even in oxygen poor environments. But if you know, there's many that don't exhibit this red color as well. These larvae will then evolve into this pupa stage that you may see floating around in some of your samples. They just kind of float on the water surface until it's time to emerge. And when those emerge at the water surface, you get another generation of flying adults. The next thing that I'll talk about is a little bit different life cycle that's represented by the mayflies, which of course we see lots of those around here, both on our inland waters and on the Lake Michigan. Again, here's your familiar aquatic adult or terrestrial adult mayfly that we see. And again, laying eggs on the stream surface or near the stream. But they will then hatch into what we call a nymph, not a larvae. And we call it a nymph because they don't go through a pupa stage. So they don't have a cocoon or something like we see with the midges. They'll spend time, depending on the species, in the stream as aquatic nymphs that actually look quite a bit like the adults. Nymphs are famous for that. They look a lot like the adult, they just don't have the wings. And then mayfly nymphs will emerge as the subimago first, and that looks a lot like an adult, but they actually have one more molt to go, and they'll molt one more time after they've hatched. I've actually had that happen on my sleeve before. You know, you're out on the shoreline, and these mayflies are hatching. They'll land on any potential surface, including you. They'll shed a skin one more time, leave that on you, and then they're adults, and they go looking for a mate at that point. So those are a couple of the common life cycles that we see. There are a few aquatic insects that are aquatic their entire lives, and that includes some of the beetles and true bugs. But most of them have a terrestrial stage as well. So let's look at habitat and think a little bit about how living in a stream can be kind of challenging for an insect. So current is a big issue, right? So having the stream moving and that force is always against you. It's like if you lived in a wind tunnel all the time, you need to adapt to that because you're going to spend a lot of energy just trying to stay put. And so the same thing is happening to our invertebrates. So, you know, some invertebrates, their approach is to lay low. And that's what we're seeing here. So this is a water penny beetle larva. They are called water pennies because they look like little coins. They're smaller than pennies, but they're almost like an armadillo, and they'll just be really flat up against the stone surface or the substrate there. And that way the water just flows right over the top of them. It doesn't knock them away. Some of our mayfly larvae, like the one you see on the right, that flathead mayfly nymph is also very flat. You can see, you know, it almost looks like it was run over by a steamroller. But that is their adaptation. They lay low and they stay low enough that they're actually, the force of the current doesn't remove them from the substrate or stones that they're hanging out on. Another way to stay put is to hold on really tight. And that's what our black fly larvae do. The Similiidae that you see here, there's a bunch of them attached to a rock surface, and they have down here at their tail end, they have a little circle of tiny hooks, and those tiny hooks they use to hold on to the surface. They'll hold on to little filaments of algae or onto the stone itself or onto the dirt particles to hold on, and then their heads actually stay up in the water current, and we'll look at that in a minute more closely, but they have ways of holding on tight. And that lets you think about when you're out there with your net and doing some sampling that you may have to, you know, definitely do some disturbance of the stream bottom to get those invertebrates to come loose. Another thing you need to do in your habitat is hide, right? So most of these invertebrates are prey for something else, either larger invertebrates or fish. Amphibians will eat invertebrates as well. And so, you know, they'll either hide in amongst the stream bottom material and the plants and the wood. So again, another reason to be thorough in your sampling. Or they may build a hiding place like these case building caddisflies that you can see here in this image. You know, building a case for itself. The invertebrate is actually about this long. They look kind of like a caterpillar, but with some longer legs here at the front. And they build this case. And the cases are actually useful for identifying what kind of caddisfly you have, because each caddisfly genus or family builds their own kind of distinctive shape of case. And they use the materials around them, so it really helps them camouflage themselves as well. So this is just one example. There's many kinds. So it's always good when you're looking at your sample and picking the bugs out of it to look for that twig that seems to be crawling around on its own. That's likely a caddisfly in its case. All right, the next step of living in an aquatic habitat for these insects is finding food. So here's our black fly again, and a horrifying, you know, electron microscope photo of its head. And this is the part where you see, we've seen this image before. This end is hanging on to the substrate, and up here you've got the head that's up there in the water column and water flowing by it. And the black flies have these intricate fans on either side of their head, and they're using those to catch any kind of particles or food bits, algae, plankton, whatever might be flying by, and it gets snagged in those fans, and then they can bend them down towards the mouth and nibble on whatever good stuff is there, clean it off, and then up they go again. So that's how they're gathering food without having to move around. They can stay put right there, not get swept away, and let the stream bring the food to them. Caddisflies do a very similar thing, but they have to build a net. So this is an example of a caddisfly that doesn't build a case, the hydrocychids, which are pretty darn common. And this nice sketch of the tiny net that it will spin, making silk, basically, like many other invertebrates do. And it uses that net to catch things that are floating by, and then it'll crawl out of its shelter, nibble on the tasty morsels that have caught on its net, spit out anything that's just fouling the net, and then go back and wait for its next meal to arrive. And I threw in an interesting picture here on the left, which is actually an image of the nets that are woven by different species of caddisfly. And the first thing you might notice is that the mesh size is different. And that's a really interesting thing, because as you might imagine, if you have a stream that's really got a lot of silts and sediment being carried into it, if you're a caddisfly that weaves a very tight net, it's going to fill up with silt all the time, and it's not going to be a good habitat for you. And so the species that weave these very fine mesh nets typically aren't found in muddy streams or streams that get a lot of erosion or sediment in them. What you tend to find is caddisflies that will weave these larger mesh nets that don't get clogged constantly. However, if it gets silty enough, you won't have any caddisflies at all, because they can't efficiently eat using their nets. And of course, there's some that are predators. And you were asking about what your favorite invertebrate is. I do have a soft spot for the Helgramite, this guy right here. They are very efficient predators. This is one has actually got the head of a stonefly in its mandibles right here. They are very effective predators on smaller invertebrates. The water striders we often see, they are predators as well. Many of the, and that's a type of true bug in the order Hemiptera. They have this sucking mouth part that they use to pierce their prey and suck the juices out. So they're actively preying on things, as does this toe biter, another true bug that will hunt smaller invertebrates for food in the stream bottom. And of course, dragonflies. Okay, these really are my favorite. The whole group, the dragonfly nymphs are a fascinating group, dragonflies and damselflies. And they tend to kind of sit and wait for prey to come by. And if you've never seen the jaw structure on a dragonfly nymph, it's pretty fascinating. This is actually an extended where the jaw has been pulled out. The lower jaw can actually shoot out and drag prey, unwitting prey that's wandering by, pull it into its mouth. So that's what's going on here. Here's a larvae that's got their lower jaw retracted, and it actually comes all the way back to here. And then when it sees prey, it lowers it and then shoots it out and drags the prey back in. And if you're really passionate about invertebrates and dragonflies and you get into identifying them down to species, the shape of this lower jaw can help you identify them. You can see this one's got a really flat lower jaw. This one is scoop shaped. It's a very helpful characteristic for identifying which kind of dragonfly you're looking at. And then, of course, as adults, the feeding structures can be very different. Now we're above the water, we're terrestrial now, and there's many of our adult aquatic invertebrates, when they are terrestrial and adult, they don't feed at all. Mayflies do not feed as adults. They are on a singular mission to mate and lay eggs. They don't even have a functioning mouth or digestive system as adults. And the Dobson flies are the same. The Dobson fly is actually the adult of that Helgramite that I showed you before. That very efficient underwater predator does not eat as an adult. The opposite is true with our dragonflies and damselflies. As adults, they are very effective predators, as many of you know. They are, people love them because they learn that they eat mosquitoes and gnats, and they catch their prey on the wing, flying around and actually holding their legs in kind of a basket beneath them and swooping around and snagging smaller flying insects in the basket they make with their legs and then enjoying them after that. Another challenge that our invertebrates face in the water is getting oxygen. How do they breathe? And so there's a lot of different ways that different invertebrate groups get oxygen, and they are very closely tied to water quality in streams. As you may know very well, the cleaner the stream, the colder the stream, the more oxygen is dissolved in that water and available for invertebrates. As streams warm up and as they start to carry a heavier silt load, as they start to slow down, so warm, slow, siltier streams have less oxygen. And so That's where stoneflies, like we're seeing here, are really good indicators. They get oxygen primarily by diffusing oxygen. It gets diffused or absorbed through their skin and through the little very thin and very basic kind of furry looking, hairy looking gills that they have. Some of them have at the base of their legs. They're very primitive gills. They're not very efficient. Diffusion is not a very efficient way of getting oxygen. So as soon as a stream starts to become polluted, too warm, slows down, you'll lose your stoneflies because they can't get enough oxygen through their very basic mechanisms. You'll tend to see bugs that are a little more efficient at getting oxygen. These are mayflies, another example of a group that has very obvious gills along the sides of the abdomen, and the shape and appearance of those gills can be, again, indicators of what species you're looking at. And so they use these gills, and if you have a live mayfly larva, a nymph in a pan, you can often watch those gills because they're constantly moving. And that's moving water across that surface of the gill, allowing them to get more oxygen more efficiently than a stonefly can. So they're a little less sensitive to declines in oxygen than stoneflies are, but they're still fairly sensitive. And then another way to get oxygen is to rely on surface air, going up and getting air. And so that's what a lot of our aquatic beetles will do. You'll see in these pictures, you can see carrying this bubble around kind of on their tail end. They'll swim up to the surface and basically grab a supply of air from the surface, and they will swim around with it like a little scuba tank. And so they don't care if the stream is completely polluted, has no oxygen, barely in it at all. They don't care. They can go up to the surface and grab air, oxygen from the air. Plastron is another example, and that's what these beetles that are quite common, the elmid crawling beetles, they actually hold a supply of oxygen underneath their carapace there, and so they have a little scuba tank as well. They don't have to refill all the time. Other invertebrates will also rely on siphons and tubes and different kinds of basically snorkeling equipment. This is a mosquito larvae, and here's its little tube that it uses. You'll often see these hanging from the surface of a stream by their tail ends, and that's because they're breathing air and not getting oxygen from the water. And maybe you can see this guy here. This is a water scorpion hanging down, and it's related to things like praying mantis and walking sticks. And they have a siphon that they have on their tail end that they can use to breathe oxygen from the air. So these bugs will be much more tolerant of pollution. All right, that's it. So I hope you enjoyed that little kind of whirlwind cruise through some of the interesting adaptations that our different stream macroinvertebrates have, and that it gives you a little more insight into their lifestyles, which will help you find them and be more efficient when you're out there doing your monitoring. And I'm happy to take questions, either being able to turn on your mic and ask, or typing things into the chat. And of course, you're welcome to reach out to me. There's my email address there at the bottom too. If you have questions, you can ask me. And of course, Caroline and Heather are very knowledgeable about the invertebrates as well.
I have a question, if I may.
Sure.
I guess it's been since last fall, but when I was looking at your stonefly images, I started to wonder if I might be misguiding identification for myself and my students at times with mayflies. I've never really had that thought before, but there's one stonefly image in particular that I was like, ooh. So I just wanted to bring that up and ask.
Back up. Did you want to look at it again? Was there something?
Yeah, I guess so. Besides, I think of mayflies 3 tails, stoneflies 2 tails, right? That's the big identifier.
Mm-hmm.
Okay, and then I think that's it. I just needed to work through that in my brain.
Yeah. So that's typically it. There are a few mayflies that the center tail is really, really short, and so they can look like they have 2 tails instead of just 3. Another thing to look for, and I know you have to get in there kind of close, but stoneflies, in addition to just having 2 tails, they'll also have 2 claws on every foot. And mayflies just have one. So they have stoneflies have a pair of tails and a pair of claws, while the mayflies do not. They just have a single little claw on the end of each. Now,
That's super cool.
Your handle lens to see that, unless it's
Right.
a big one. But and then what you'll also see, not in all the stoneflies, but in a lot of them, they will have these tufts of gills at the base of a lot of their legs. That one has it. That one has it. This genus doesn't. While mayflies, their gills are all on their abdomen, not on the.
Okay, excellent. Yeah, I think it was the gill mention that made me think like, oh no.
Stoneflies have gills? Oh no.
Yeah. Yeah.
They
So
look
thank
a lot
you.
different. Yep.
Joe, is it also true that stoneflies usually have more pronounced antennae?
Usually, yeah. The mayflies tend to, most of them have finer and maybe a little bit shorter antennae, while the stoneflies tend to, the ones we have around here, tend to have more prominent antennae. So yeah, that's another kind of thing you can kind of catches your eye when you're first looking at them. Absolutely. I got a question in the chat about distinguishing New Zealand mud snails from other snails you might find. They're tough. They're really small, so that helps. And you know what I tend to do, I've actually, to be honest, I have never found a New Zealand mud snail in the field. But I haven't looked very hard. Like I said, the last few years since it's become a problem, I've been working more in lakes than I have in streams. And so, you know, there are some really good resources online, especially through the state of Michigan, to help you identify those. And Oakland University has put together a little tip card. I think I gave some to Heather on how to identify them and tell them from others. So I'm probably not the best person to give you tips for the field, but you know, you definitely want to be alert to any small snails that you see in the stream, because that could be something worth looking a little more closely at.
Hi, Joe. Rod Rebant.
Hi.
I'm curious, do you have a favorite textbook or a reference for macroinvertebrates?
Yeah, and if you don't mind, I'm going to step back and I'm going to disappear because of my virtual background, but I've got a book on my shelf that I want to show you. So I'm going to grab it real quick.
Thank you.
Okay, this one right here, and actually I'm going to turn off my virtual background for a sec because you're not going to be able to, if I hold it up to the camera, it's going to disappear. There we go. So right here, Freshwater Invertebrates of North America, a guide, and the author is Voschel right there, Reese Voschel. You can get it, you know, anywhere books are sold. Everybody just says Amazon, but you know, there are other places to get books. But this is a really great reference and has really good descriptions. And also, let me show you, it's not very big, so it's, you know, easy to keep with you in your pack or whatever. And then it's got really nice drawings of the different invertebrates and descriptions of kind of key characteristics. So yeah, A Guide to Common Freshwater Invertebrates of North America by Voschel is a really nice field guide.
Thank you.
Mm-hmm. Well, thank you so much for having me today, everyone, and thank you for your volunteering. We're really excited. You know, I'm continuing to have strong involvement with the MyCore program, and you know, always interested in feedback from both program coordinators and from individual volunteers about, you know, what resources we could provide and how we can support the great work you're doing. Because as Heather and Caroline already mentioned, this program is vital, both locally and on the state level and beyond. We watch the use of our database where all of the data gets entered, and it's actually really amazing the number of people that look at that data and are interested in it. Everything from teachers using the data in their classrooms to researchers using it to choose study sites, or college professors that are using, able to use real data to teach their students about stream ecology and lake ecology instead of just, using a made up data set or taking something from that isn't local to Michigan. It's a really valuable, and of course the state uses it for tracking water quality all around the state. So thank you for putting the time in and going out and doing it. I know it's a lot of fun, but sometimes it feels like, oh, there's so many other things we're trying to do in life, but it's really appreciated, all of the work that you do.
Thank you, Dr. Lattimore.
You're welcome.
All right. So I think, Caroline, next we'll break into our breakout. If you want to get a sponsor, please contact us and we could, try to think about some sponsors that might be worthwhile to reach out to. And then folks that usually pay their program fee, we have like a $50 suggested annual program fee. But of course, you weren't monitoring in the spring, so we're suggesting $25. But really, it's no big deal. We know that this year's been super challenging for many people for many reasons. We're not going to hold you accountable to that annual program fee this year if it's not going to work out for you. Any questions?
Nope.
I love how my computer is not going to allow me to advance the slide. Can only take so much. You might have to.
I feel you, Heather.
Can you see my blue swirly of like death?
Yeah. As a teacher who's trying to do this all the time, I feel you. So hang in there.
I'm gonna try something else. Okay, can you see this?
Yes.
All right. So I just did a few of our common invertebrates that can be tricky to identify, and thanks to Dr. Lattimore for going over a lot of them. But these are our common caddisflies. So we know that they're the free living, and then they're the case making. So the case making, as Dr. Lattimore was stating, you'll see either what looks like a stick or a rock that ends up like walking away. So it's good to like stare in the bottom of your white identification tray for a few minutes, and you see something moving, pluck that out. These are case making. They look like they're essentially crawling out of like a sleeping bag or a turtle shell. And again, just kind of like trying to stare in and see if anything's moving. That's a stick or a rock. A good way to find those. Those are our more sensitive caddisflies. We have a number of other caddisflies that I find all the time in our stream sites. The most common is this common net spinner. I'm sorry, I spelled that wrong, but it curls up in like a C shape. That this image here in the bottom left is, we see this kind of shape of how it curls up all the time. It's got some, it has like these three hardened plates near its head. That's another good indicator. And then it's got a pair of, let's see, 3 legs up here. And then they have pretty bushy tails, or, the technical word might not be tail, but you all know what I mean. Next, we have this one that we've been finding a lot that is really fuzzy, and the fuzz is kind of orange, and it's a real bright green. The book that Dr. Lattimore held up, don't have it with me here, is the book that we use for identifying invertebrates, and I have a number of copies that folks are welcome to borrow if they need to as well. But that has a really nice image, and that I can't believe that how important it is to see the color of that image, because when you see that bright green caddis, you're like, yep, that's definitely this caddisfly. And then finally up here we have the fingernet caddisfly. They're almost translucent. You can see kind of the insides. They kind of have an orange yellow hue. You can really only see 2 hardened plates near the head, and they don't have super bushy tails, but the they're kind of strange in that they're super translucent. Any questions with the caddis, the common, the caddisflies that you may encounter in the stream? Folks are pretty good at identifying them. Next are the mayflies, and Dr. Lattimore talked a little bit about this. They're super diverse. This is an image that I took from macroinvertebrates.org, which is an awesome website that goes through a bunch of different morphological features for each type of invertebrate that we might find. But this just illustrates how diverse these insects are. Most of them have 3 tails. There is a small common, the common small minnow mayflies. They have 2 tails, and we see them a lot in our streams, and folks often misidentify them as stonefly, but and you can kind of see this one. I think is one of them. Do you see these 2 tails here?
Mm-hmm.
Do you see where I'm pointing? Okay. Those 2 tails seem to originate from that same spot. You don't really have that gap that you see, that really pronounced gap in the tail in the stoneflies. These common small minnow mayflies also have a really distinct swimming pattern. They like kind of go up and down, like they follow this like wave pattern. So once you identify that swimming pattern, they're usually pretty easy to tell from the stonefly. Most, or I should say all mayflies have those abdominal gills, and they usually don't have very pronounced antennae compared to the stoneflies, at least compared to the stoneflies that we're finding here in northern lower Michigan. But they're super diverse. Any questions at all about the mayflies? Okay, here are stoneflies, and then you can see in some of these images that really distinct like gap kind of where that tail originates, really distinct here and here, and you can see how a lot of their antennae are like super pronounced. Sometimes we find really big stoneflies as well. You know, if you find a really big, juicy, 2-tailed creature, that looks like a mayfly, it's likely a stonefly. Although they obviously are very small as well. And then another one that's sometimes confused with both the stonefly and the mayfly is the damselfly, because they have 3 tails. They wow, this is a mistake, so sorry about this slide error. Don't read anything right here. Cover that up, pretend you don't see it. The damselflies have 3 tails, always 3 tails, and their tails are really wide. They almost look like a fan blade. They're never like the hair kind of width of both the stonefly and the mayfly. So that wide blade of their tail is a sure sign that you have a damselfly. They also are like a little bit more gangly, like, is that even a word? They just have like these long kind of awkward arms that can be really distinct when you find them. And oftentimes they're a little bit larger than some of our smaller mayflies and our stoneflies. Questions at all about stoneflies and damsels? Okay, one thing that I noticed is that sometimes midge larvae are hard for folks to identify. They get confused a lot with beetle larvae or aquatic worms, but the best advice for a midge larvae is that they have very jerky techno-dancing moves, like super jerky. Once you've identified the way that they move, you can't unsee it and you can't not identify it. They just have really jerky movement where they flip. You usually find one, you find a million, so it's very uncommon that you're gonna find like one midge larvae in your sample. They're oftentimes really, really small, so sometimes they're hard to see. They're almost in all of our samples at all of our sites. They're just really tolerant and they're super ubiquitous. They are they're too short to curl up on themselves. So if you find something that's like tying itself in a knot that looks like a worm, that is some sort of aquatic worm. That is not a midge larvae. Questions about midge? And then, of course, those bright red ones are the blood worms or the blood midge. Okay, is there anything that anybody wants to talk about or any questions?
Will we have access to those pictures? They're really helpful.
Yeah, I can certainly email them to all attendees.
Okay.
Yeah, I was going to confirm. I noticed that the session's being recorded, but I was curious if we could just get access to the slides as well from tonight.
Yep, I will make sure that happens.
Thanks.
Yes.
Heather, can you remind me again what the collection dates are?
Yes, that's a good thing for everybody to know. It opens up on Saturday, October 3rd, and it'll run till Saturday, October 17th. So anytime during that period, you're welcome to sample. We're really hoping to get samples back by, hopefully folks are going to drop off on October 19th or October 22nd, but really shooting to have all samples back by the end of October so that we can do our, so we can promptly do our verification. Another reminder that we know that the biotic index is based on abundant or rare, or those are essentially the two categories if you have an invertebrate that's present in your sample. The abundant is 11 or more, but we ask you to identify 15 of those, or preserve 15 of those specimens, just in case you misidentify. So if you had, 12 mayflies, but 2 of them are stoneflies, it would bump it back down to the rare category. But if you actually had more, 15 gives us a little bit of a buffer. And then, of course, just preserve all the rare ones that you encounter. Any other questions? Any other info?
Oh, go ahead.
Oh, this is, and this might be better at the end or just with Heather. I was just going to ask about the data sets and that sort of thing for my core. This is more of like a teacher question. So why don't you go ahead?
Okay, I just wanted to clarify on that last point, Heather. So you said, collect 15, but you also mean collect like 15 scuds and 15 isopods and...
Yep, even the most commonly identified. We're going to encourage you to collect 15 of the commonly found inverts.
Okay.
Just in case. But please don't give me like 45 scuds.
No.
Just joking, I'll count every one of those cute buggers.
And could I just request to get habitat? I'm not due for a habitat survey on Yuba, but if I'm able to get out to some other locations, then if you can give me a blank one, then I can always photocopy it.
Yeah, absolutely. I can email that to you. And I'm hoping, Mike, that you and I could talk about some sites that are left unmonitored that you maybe want to pick up or help me with, or help maybe Jim and I with.
Okay.
Good thinking.
Any other questions?
No.
Tani, are you feeling comfortable sampling weaver by yourself this season?
Actually, I said that I would help Rod do one of his, and he's going to help me do weaver.
Oh, wonderful. Then you're in good hands.
I know. Isn't that very nice of him?
He is a very nice guy. Good, good. That'll be great.
Because no, I didn't feel comfortable.
Well, it's a lot when you know, it's been a year since you've been out to the site and you've only been out to the site once or out sampling once. And Rod, do you know if your team's still going to do west branch of 4 Mile Creek?
Yes. There will be 3 of us doing that sampling this year, so we won't get the complete set, but put as many people in the water as we can.
Okay, wonderful. Good to know. Okay, Jenna, did you want to ask your question? You might be the last.
Yeah, I was just wondering if there are any data sets that come to mind specifically that would be interesting for me to share with my students for checking out. You know, I take my students out in the stream and we do essentially the MyCore protocol, but we aren't reporting it. So I'm going to look into it because my lab time is a little different this year. So I might even be able to get registered. But then on the other end of it, I was wondering if in your work, if there's anything that comes to mind, like site-wise, that would be like, oh yeah, check out, you know, this site for the data that have been collected there.
Yeah, you know.
And if not, that's totally okay.
Yeah. So you can always peruse if you go to mycore.net, there is, there's also a lake monitoring program that's ran by MyCore, and then there's a stream monitoring, but you can go into the stream database. It's on the right side of the screen. And I think that database is still live. They're doing an overhaul of that database. So it's going to close like for a period of time, for a few months while they move it over to a new system. But the last time I checked, which was maybe a few weeks ago, it was still accessible. But you can go in there and if you haven't played around that database, it's really interesting. You could like, if you were interested in lake data, you could like go by parameter or go by county or go by, you know, time sampled or like year sampled. But one of the things that might be interesting for this group to look at, and maybe it wouldn't, but you know, Kids Creek is well, you're on Kids Creek. You're on in the impaired section of Kids Creek.
Yeah.
It's impaired for aquatic life. As a compromised aquatic life, so compromised invertebrate community, and mostly we think due to stormwater and changes to natural flow. We've been floating around some other ideas, and Sarah Uren, who works at the Watershed Center, she's our program director. She's really like the queen of Kids Creek, but she's been like tossing around some ideas, like perhaps it's related to road salt, perhaps total chlorides are too high, like they're in toxic amounts that's hurting some of our burrowing insects. So we have a lot of questions about Kids Creek, especially in the area that you're at. But it's interesting to look at how Kids Creek data is different in different segments. So like,
like, well, in that section that got lit or whatever restored recently.
Yeah, we have that daylighted section. That's where Frank and Joan are. Yep. That is an interesting site, and that's been better than the main branch, I would say, historically, or in the impaired section. But as Frank and Joan both know, that's received some sedimentation over the last couple of years and filled in with sand. But like Kids Creek Trib AA scored pretty high, and that's kind of this tiny trib feeds right into the main branch. And then it's also been higher kind of upstream, like even north, or I should say south of, upstream of like coals and like Great Lodge and stuff. There's like some higher quality habitat. So that might be something interesting to look at. But I'm trying to think of what else.
No, that's cool. I think that I've played around with it in the past, but it's been long enough that I've forgotten whatever I did. And so I just thought like, oh, well, since it came up, I'll just ask today if there's anything that came to your mind immediately. And Kids Creek did come to my mind, both because I work on it and because there are these sections that are, you know, had restoration occur on them and then are still like suffering from channelization, whatever
Yeah.
else.
Yeah. Yeah, it's an interesting one.
Yeah, cool. So that was helpful. Thanks.
Yeah, maybe other volunteers have suggestions too. Anybody have interesting data from their stream sites?
One thing I was curious about was if there might be just thinking about the reference to the change in sampling over the season. Isn't that something like a seasonal succession thing too, where you might have nice diversity in the spring, but then in the fall you might just get a ton of scuds because of all the leaf litter and their populations have grown? Or do you not see that seasonal change?
That's really an interesting question, right? Like if there was a seasonal change in scores, like if certain streams score higher or lower in different seasons. Not usually. So most of the invertebrates that we're looking at have at least 2 life cycles in a year. So usually like early June, late May is when we see some of the larger invertebrates in that juvenile stage, like getting ready to emerge as adults. So we pick that time of the year because they're easier to see. And then again in kind of mid-fall, most of them are like ripe again to emerge as adults and are larger to see. So there are like numerous life cycles in a given year. And I think most of the inverts have 2 life cycles, and that's why we pick the spring and the fall. And actually we don't have much, you know, my course protocols say we have to sample in the spring and the fall, and there's some latitude in terms of, like, I know that Carolyn's team is sampling at the end of September, and we always sample the first 2 weeks of October. So there's some slight variation there, but generally that's how most of these programs are. Some of the larger watershed groups across the state have like a winter stonefly sampling event. Apparently stoneflies get really big, and maybe some of them even emerge in the winter. So that would be a cool thing to add someday if there was a lot of community interest.
That's super fun.
Well, we do on inland seas on the school ship, we do see quite a variation in midge larvae seasonally.
Oh.
So like late July, August, we get very few in the dredge samples, and then it picks up again in September. So
kind of that spring and fall.
Yeah, and then they only sail May to October, so we don't really know what happens in the winter. But,
right.
But yeah, we'll get like 40 or 50 in a dredge sample in the spring and May and June, and the same in September and October, but probably only 5 or 10 in a sample in August.
All right, anything else anybody wants to discuss?
All good.
All right,
feeling good. Glad we get to sample.
Yeah, great. I'm so happy that everybody is feeling comfortable and excited to get in the stream. We're excited to have y'all back, and we are excited to see some of you in person during our pickup and drop off. I'm sorry if I'll miss you this season, and looking forward to being able to actually get in the stream with you again, hopefully next year.
Thank you.
Be sure to send out slides and a link to this presentation once we can get that finalized. And any questions, let me know.
Thanks very much.
Thank you, Heather.
Thank you all.
Thanks, Heather.